Okay, thank you. So my name is Lana. This is the title of my talk. It's kind of long and ugly, but at least it's very descriptive, so you know what you're in for. But actually what I want to talk about is this. When we think of evolution, we think of at least these three characteristics in a system. Self-replication, so making copies of yourself, with mutation, so not exact copies of yourself. And then selections, so a force that makes some mutations more fit than others. And if you like open-ended evolution, you also would like to see something like exponential something. So exponential increase in complexity or in novelty. And my talk today will be about a system that has all of this except selection. So the actual title is Almost Evolution in Neural Cellular Automata. Before starting the talk, I want to give you some links. So here you have a link where you can leave comments and reviews about the paper. Very interesting website called Chaos. Some people have already left reviews. The code is available at this link, which is, the link is also in the paper as a collab notebook. And then I made some kind of game that you cannot see, okay, related to this paper. So you can access this at this QR code. And basically you have an egg with two genes. You change the values of the genes and you're trying to obtain a hybrid between a fish and a lizard. That's the game. So sometimes you get kind of greenish fish, you're almost there, but you can also reset to some known values, like the lizard, and then you run it. And you get a lizard. So try to get a hybrid, try to find the values, harder than you think. So we had at least 15 people explain what cellular automata are in the last 2 days, so I will be very quick. We have a grid, we have some cells. The cells have states, and the state of the cell will change according to some rules and to the state of their neighbors. And it used to be that we made these rules by hands, but thanks to Alex, we now have a very convenient way to learn the rules. So you only have to decide the initial state and the final state, and then the neural network will learn the rules to go from initial to final. It's pretty robust. It feels more lifelike than other automata because it has some interesting properties like self-repair. But most importantly, it's not hand designed. So you have this neural network that will learn whatever you want it to learn, or at least try, which means that we can now have very complex rules because we don't have to think about them and write them by hands. So you just decide, this is my first state, and this is my last state, and please go from here to there in a number of steps. And what I wanted to know was, can we get some interesting self-replication? And strangely, there doesn't seem to be a lot of work about self-replication in neural cellular automata. And I think that's strange first because the first cellular automaton by von Neumann was created to study self-replication. And also because it's very easy to just specify what you want to the system. I want to start with this bacteria, and then at the end, I want 2 bacteria. And that's all you do. And you get something that looks like self-replication. So the bacteria kind of divides, and then you end up with these 2 fuzzy bacteria. And that's where the problems start. So you can do this. You can have 1 bacteria, and they get 2 bacteria. But then the system has learned that it should replicate this exact image. So this fuzzy thing will not replicate. You will just stop here. What you actually want is 2 bacteria, and then this one should also divide, etc. So to get that, I'm sure there are some very beautiful ways to do it, but this is how I did it. It's not very pretty. You have some, when you train the cellular automaton, you have a batch of input data, and you get the output data. And this is the first training step. At the next training step, instead of having these beautiful, clean bacteria, I will copy the output here and put it as an input. So I trained the system to replicate even this fuzzy, ugly bacteria. And then now it works. So if you just run the system after learning, you get something that looks like this. So it divides, and then it divides again. And because of the boundary conditions, it stops dividing. So we want more division than that. So what I do is when the bacteria divide, I randomly select the top or bottom bacteria, and I transplant it to a clean grid, and I just repeat this. So select, cut, paste somewhere else, and let it divide. So now it's dividing for longer. And we can also see some interesting thing happens where, for example, here, it's very clear that the bacteria has mutated somehow. It has 2 nuclei instead of 1. So I got interested into these mutations. And I thought if I want to study mutations, it's better to have something that is like DNA rather than just phenotype. I wanted to decouple the development, so from DNA to the organism, and the replication. So the organism should also, so I choose this fish, there's an egg becomes a fish, and then the fish must also lay an egg. And now the next issue is, do you have 2 steps? You want the automaton to learn 2 things. First, development, and then replication. So again, I'm sure there are some very beautiful ways to learn 2 targets with the automaton, but what I did was just this. First training step, learn development. Second training step, learn self-replication, and then repeat for some thousands of steps. And you can really see when you look at the loss of the network, at first, the loss for the 2 targets is different, and then it kind of converges, and you get something that self-replicates. So if you cut the new egg every time and put it in a clean grid, you get some dynamics like this. So the fish, it kind of, it's not a real motion, but it goes away and then it lays an egg. And you cut the egg and you repeat this for a few 100 generations. If you don't separate the egg from the organism, you get something like this. I wish I had time to talk about this. I think it's a very interesting problem because everything becomes a fish. Any little pixel that you put in, it will become a fish. You can copy paste a bacteria emoji, it will become a fish. So that tells me that this is very far from life. It's more like the fish is a laws of physics of this world. So I looked at the successive generations. This is generation 0, 1, 2, etc. At generation 98, you can see something interesting. The fish has lost a stripe. It's supposed to have 3 stripes, and it has only 2. At the next generation, it kind of like recuperates this stripe, and then at the next generation, it has too many stripes. If you look at the difference between generation 0 and all of its descendants, mostly the difference is about the same. And then around here, some things happen. So you have a stripe that becomes forked, and then it recovers and becomes a bit strange. What's interesting here is that in artificial systems, it's often very hard to get mutations that look like biological mutations. But this type of mutations, like losing a stripe or getting a fork, really happens in biological systems. Now, when you look at, instead of just the difference between generation 0 and all of its descendants, you look at the average difference between parents and children, or grandparents and grandchildren, and you do this average for 100 generations, you get a curve that looks like this. So kind of straight, maybe slightly exponential. The difference between one organism and its descendants just increases over time. But sadly, they are still all fish. So it might look interesting, but actually the scale is so small that whatever you do, even if you impose some selection, like choose the bluest of the children or the biggest or the smallest, they're still all fish. You cannot really get away from this. So what can we do to get something more interesting? Well, you can try to train the automaton on 2 different organisms. So my idea was maybe if I train on 2 attractors, I will get more attractors for free. So fish, lizard, and maybe some hybrids in between. So to do that, I really just make the grid a bit bigger, put 2 eggs with different information in it, different DNA, and then train the automaton to learn development and then self-replication. The results look like this. So you start with a kind of a fish, then it becomes a bit greenish, half lizard maybe. And so you just continue like that for a few generations and you get these types of dynamics. So fish slowly becomes different, it becomes very small. What's interesting here is that you have a very small egg and a very small fish. And then it becomes a lizard. And then it goes back to being a fish. I don't know if that's creepy or fun, but I like it. The actual interesting thing is when you look at the same figure that before, but for these 2 organisms, cellular automaton, the DNA difference is about the same order as the previous model. So for the only fish model, you had the maximum difference of DNA of 0.1. For this 2 organism model, you get about the same. But when you look at the phenotype, for the only fish model, the maximum difference of phenotype was 0.04. And here you get 1 order of magnitude more difference. And when you look actually at like the shape is kind of exponential, the phenotypes are also more interesting. So you get some small, big, no face. And it seems to continue for a while, but again, you're kind of stuck with just fish or just lizard. So in conclusion, if you train a neurocellular automata just for self-replication, but robust self-replication, you get exponential genetic drift for free. Sadly, because we are using a deep neural net, they are made for convergence. So the NCA will tend to converge to just this attractor with some mutations. If you want divergence, if you want something that is exponential or open-ended, you must force it to happen. I'm not sure if you train the automaton on, say, 100 attractors, do you get 200 attractors for free or not? I'm not sure, but I don't really think that's the way to go, to be honest. And I thought I wouldn't have time, so I didn't talk about modularity, but since I have time, I can talk about it. Okay. So one thing that could happen here, that doesn't happen, but it could happen, is that you get something that is close to paint by numbers. So all of the genes might give you a fish, and in just one little change will give you a lizard, and there is no modularity. So to avoid that, another experiment that I did, it was having several seeds that have exactly the same information, but look, the position is different. And so the whole, information about what organisms should be created is encoded into space instead of being encoded just by the DNA. So when you do that, you get modules that start developing the same. And then when they meet each other, they use this spatial information to differentiate into different things, which is also what happens in embryogenesis. And that's all. Thank you.